I have to give a little bit of a disclaimer from the beginning. My background is a funky in-between place, right? So I started out as a child neuropsych doctoral student, and along the way a brilliant professor converted me to the statistics side, whatever you want to call that, light or dark or whatever. And so when the kind of methods that I'll be talking about here, I try to emphasize what the clinician needs to know. And that tends to be a funky mix, in my opinion, between some of the N-of-1 designs that we heard yesterday and statistical and methodological rigor. So I hope, just be open to this being a little bit different. I was also told that I can't put formulas up here, so I'm going to try to not get too complicated in the math. David couldn't resist putting one up at the end, and I have to say, I felt like giving into the temptation, but I didn't do it. So what I'll be doing is communicating verbally and with visuals. And I think that to communicate these well, I'm going to spend a good part of my time on a couple of examples to show what these kind of techniques can accomplish. One quick point also, the word idiographic starts with an I, starts with the one, the individual. That's in contrast to nomothetic approaches, which start with an N or sample size, and really emphasize, taking from the sample and learning about a population. Here, in the idiographic techniques, they emphasize learning what's happening within an individual as they're exposed to treatment. Okay, and as a referral to what we'll be doing next, or how this fits rather, John and Mary Catherine gave examples of N-of-1 types of studies and multiple baseline designs. This starts with that same sort of foundation and builds upon it. So if you kind of keep that context in mind, that's where this is coming from in part. Just as a quick outline, I'm going to try to sell you some of the strengths of ICTs and their potential for home visiting. I'll describe exactly what ICTs are and perhaps how they differ from other approaches, and when to use ICTs and when you don't want to use ICTs. There are definitely places that they don't belong. And then I'll spend a good bit of time on the illustrations, as I said. So some of the needs that idiographic clinical trials meet. They are adept for situations when you have small samples or small populations. This is not a small issue. There are something like 7,000 rare diseases in the United States that you cannot run randomized clinical trials on because there aren't enough people to do it with. So that would be an example of a small population. Small samples might be like when we heard Mary Dozier talking yesterday during that tweaking process of understanding exactly how an interventionist can best communicate things most effectively to the people that they're working with in that training piece. So she couldn't go out and recruit 400 and some people to do that kind of a study, right? She had a handful of people. And that's where these kind of methods can be useful. If the research has to require doing things in the field, in other words, you can't follow rigorous randomized clinical trial protocol necessarily, but you still want to do rigorous research, this is an option for doing that. These methods are really handy for picking out active ingredients or processes that are occurring within people. They fit well within the precision treatment notion. And then in a couple of the examples, I'll describe how we can approach learning what works for whom with these methods. Also, these are processes that can be used for rapid program evaluation. A participant's typical time of participation ranges between 2 weeks to 3 months. And so that study process can be accomplished within a couple of years. So when you're talking about R21s, R34 type of proposals, these fit nicely within that idea or that timeline. And then these techniques dig very deeply into heterogeneous outcomes. I won't be getting into that so much, but hopefully I can communicate some of those ideas. And if we have a moment at the end, I've got a couple of extra slides that can illustrate that more carefully. So this is a process taken from pharmacy of what developing a medical treatment involves. Starting with the concept, let me see if I can use this here. Is that showing up? Yeah. So from our basic sciences, we develop our theories and ideas about what an intervention might be able to do and what targets it should be aiming for. There are quite a lot of investment that occurs at this end. We heard yesterday about the amount of money that gets invested in preliminary trials for drugs. And then there are phase 2 testing that are a little more rigorous once the treatment has been developed, tweaking it, making sure that the proof of concept notion is met, more of the practical issues, like what Mary Dozier was talking about. And then we come here to our efficacy trials. And the nomothetic approaches target this phase, right, this area, and typically phase 3. Then after that, we've got, how do you take this treatment and apply it in the real world, which may or may not use those same kind of nomothetic techniques. So idiographic clinical trials tend to hit other areas in this process. This is not an approach if you want to do your phase 3 study. Don't come to these approaches. They're not really aiming for that kind of information. They're looking at other things. So what is an idiographic clinical trial? This, in a nutshell, conceptually, it is comprised of 2 components. One is the within-subject experimental designs. Some of these you might be more familiar with are crossover design. That's used quite a bit in nomothetic studies compared to others. We heard yesterday about multiple baseline designs. There's ABAB designs, if you've ever heard of that sort of thing. And then there are longitudinal within-person analytics that are attuned for small samples in in-depth analyses of individuals. So the within-subject experimental designs include time series data. You're looking at depth and intensively within individuals over time. Each individual experiences each phase of the study. So instead of randomizing people and hoping that they're going to be equated on all their characteristics, we use the same people. That has advantages and disadvantages to it, and there's some methodological things that need to be done differently, like accounting for autocorrelation very carefully. But that's the option there.
Ty, can you speak into the mic a little more?
Yes, is that better?
Yes.
Oh, gosh, yeah, definitely. Okay. Thanks. So then these designs also are highly flexible and rigorous, so they can be overlaid onto normal treatment. One of the studies that we were involved in was with pharmacy, where people were coming into intensive care, and over half of them were unconscious, so you can't take the time to get their consent to be in as part of the study. But what was happening there was that the physicians were using their sedation drugs differently depending on what their favorite pet drug was. And so we ended up with a natural experiment where we were able to use the medical records as our outcomes data, and the condition was the physician who was on rotation at that time. We went back afterwards and got consent from the patients to be able to use their data, but there's not a rigorous protocol that we had to follow like what would have to be done with the randomized clinical trial. That's kind of an extreme example, but it can be used in other settings that way also. There's a long history of the logic and use of this evidence. Let's see. So within person longitudinal analytics, some of the advantages that we use those is that they're also focused on individuals, so they're attuned with the within-subject experimental designs. You can yield aggregates, but you also get estimates, very careful, detailed estimates per individual. And so one of the advantages to this is that you get what's called an impact. It's like an individualized efficacy. And sometimes during the intervention, that kind of feedback to a family can be therapeutic. This resolves some of the historical sources of bias in within-subject experimental design. So one of the statements we heard yesterday is how important eyeballing the data are. And that's true, and that it's a very laudable goal to have an intervention that is so powerful. You can see the difference there, but as you know, it doesn't always work out quite that way. And so what can happen when you're just eyeballing data, there are a number of known biases and sources of error in trying to interpret those data. And eyeballing data don't give you quantitative numbers to match with what those effect sizes are, and these analytics can do that. Okay, even though also the analytics are complex, the output is fairly intuitive. So you could sit down with a family with some of the graphs I'm going to show you and just show them what the results are if that's part of what you would like to do. And then they can produce efficacy-like estimates. Okay, so just as a quick reminder, this is your multiple baseline design. Here on the left, you've got 3 subjects that were in the study, and at the vertical lines is when our treatment was introduced. We'll say this is a small component of an intervention that's attempting to increase attachment. And we're counting the number of times eye contact is made between a mother and a child in an hour, we'll say. Right? So at the baseline, you can see each participant had low eye contact. But when our magical intervention was introduced, that rate of eye contact increased considerably. This is one of those nice, neat textbook examples of these kind of data that you show when you're trying to explain the exam, how to do this, right? But that staggered baseline is really important because it controls for a couple of things. One is the natural progression of a disease or whatever condition you're trying to study. Second is practice effects. If you are getting self-reports from people, say over text messages or that sort of thing, you'll see those practice effects in that baseline phase, and you'll be able to control for that. Another example is what's called historic effects, when something else happens that changes the data that you're looking at. And that's an example here on the right side. We've got the same 3 participants, but the jump in eye contact occurs at the same time point for each person rather than when the treatment is introduced. We'll say there's an outbreak in pink eye, and the parents are all looking at the kid's eyes. That's about the best I could come up with as an alternative there, but you get the idea, hopefully. Okay. So when is it a good idea to use an ICT? When you've got a small population and you want to get efficacy for it, it's not just for rare diseases. If you're looking at a very homogeneous subset of people, this could be an alternative for looking at that. As you'll see later, you might want to compare between subgroups to look for differential effects or what works for whom. In pragmatic trials, when your sample is an organization or practice, and you have few of those to work with, this is a good way to study that practice over time. Active ingredients research, this can be really helpful because you're looking at what becomes active and what these changes are associated with over time. Precision medicine, again, this fits well within that sort of zeitgeist. If you've got a particular genotype that you're looking at, usually you end up having a small sample. And so this can be powerful in that setting. EMA data or daily diaries provide data that are well attuned for these kind of analyses. And these can be useful for understanding relationship processes that are evolving over time. I'll give an example here at the end again. Some of the things they're not useful for is large population efficacy, our typical notion of how we do research with large samples and get a population estimate. That's not what these are designed to do. And sometimes that's hard, that mindset, that switch is difficult because that's how we're trained and that's how we understand things. They're not useful for acute illnesses like a cold or a flu. And then long interviews or questions are not useful here because the participants have to provide data pretty frequently. Let's see here. Some of the strengths, I'm going to skip over this because we've gotten into that, but one point I do want to make is that something when designing these to be conscious of is that you're asking participants to do a lot in that shorter time period. If they're reporting things, you want to use very few questions because they have to do this every day. One other limitation is that if you're trying to change personality or things that are hard to move, this is probably not your design of choice because it's going to take longer. One of the strengths I do want to mention is on the first 2 lines, all participants get treatment. Imagine if you have terminal cancer. Someone's invited you to be a part of a study, and there's half a chance you're going to be in a control group. That's a large part of the reason minorities won't participate in this research. I've read that the participation rate for minorities in cancer research is like 5%. So if attrition or participation rates are an issue, it's a really strong motivator that all participants get treatment in these things. It also yields an impact per participant, so they're going to get personal results from what that treatment can do for them. And that can be another strong motivator, not just to participate, but to complete the study. Okay, so here's illustration number 1. And this is an example of isolating components of intervention and what works for whom. Worked with Dr. Bethany Rafe, whose concern was trying to, wow, sorry about that if you can't hear me in the back during most of this talk. I'm having to hold this right up to my lips, which is I'm not used to. Usually short enough and close enough to the microphone, that's not a problem. So her concern was that as teens age, if they have type 1 diabetes, as they become adolescents, parents hand over the responsibility of taking their glucose test to them. Now the prescribed rate of daily glucose tests is 4 per day. And very rarely does that happen once the adolescent takes that responsibility over. So she developed a web-based program that would require very little people effort to encourage adolescents to accomplish this goal of 4 glucose tests per day, and involve getting on the web, taking your test while you're on the web, and with the camera viewing them. At the same time, the adolescents would enter the result of that test directly on the computer. But there are a couple of complicating factors here. One is that motivational interviewing has already been shown to increase the number of glucose tests they take. So she wanted to be able to control for that intervention. Then there are different contingency management ways to try to reinforce the adolescents doing this. And so she wanted to try at least 2 different ways of doing this reinforcement piece. Then it gets even worse. We've got 2 different age groups that she thinks there may be differential effects for. So to do this within a randomized trial, you end up with a pretty large sample if you're not expecting big effects, and a good bit of complication. But here with this particular study, this is how she tackled those problems. We had a true baseline period here with a pre-MI. This is called a multiple treatments design, by the way. Then motivational interviewing was given after 5 days of participating in the study. You can see at the beginning, all participants averaged about 2 glucose tests per day. With motivational interviewing, they jumped up to 3 tests per day. There's a good effect there, and that effect held strong for the next 5 days. Then came her web-based intervention, so kids were randomized into 2 different contingency management groups, so we've got kind of a hybrid design here. And they were randomized according to their age group, older and younger. So just to explain this is where we started getting treatment by age, by contingency management interactions. And so in the top 2 lines, you've got the older teens. Those who are 16 to 18-year-olds responded really well, regardless of what the contingency management was. Their benefits held over time, so that here, by the end of the study, when the treatment was dropped and we just followed them over time post-treatment, they held to 4 glucose tests a day. They met their goal. And we were able to say, you rock. Here with the younger teens, the initial effect wasn't as strong, and it tapered off quite a bit over the course of treatment, such that younger teens that got one of the contingency managements were right back where we started. So this was done with a sample of 41. These results were all statistically significant. Part of the power here is the number of observations instead of adding a number of people. And this kind of illustrates also where you can get into an adaptive mode such that if people are not meeting your clinical threshold, we'll say of, 3.5 or 4 and higher, you can introduce an alternative intervention at that point and then keep following them. This is another illustration of what the methods can do. In this particular example, Dr. Roy Cooper had designed a wheelchair for paraplegics and quadriplegics. The issue here has to do with what sitting in a wheelchair all day long has, the effect that it has on the human body. I'm trying to find somebody here who's sitting at that 90-degree angle that your chair is taking. I don't see anyone offhand. And that's exactly the issue, right? If you do that all day long, your muscles cramp up, you get bed sores, these can lead to infections. It can be actually a pretty bad problem. So what he did was to develop the power seat. And the power seat merely involves a couple of engines where that wheelchair can then be almost laid out to a supine position and relieve some of that strain on different muscle groups and give them in a different position. So they built these. He's an engineer. Really cool lab, by the way. He built these, gave them to participants, and then followed them over time where they were rating their levels of discomfort, and they were able to track the usage of these devices directly on the wheelchair itself. What ended up happening was nothing. They didn't use them, and they still complained of the same level of discomfort and problems that they had before. So they went back to the drawing board and hypothesized that so either we probably need more instruction, maybe they just don't understand how to use it, or for some reason there's not the motivation or they're just forgetting to use it. So they developed 2 interventions. One was a further instruction period, and another was a reminder to mindfully take stock of their body's comfort levels, the different areas of their body that were uncomfortable, and then use that as a guide to how to reposition their chair. They then reran this study. with 60 individuals and tested out their hypothesis that way. Fortunately, this is not a large population. They didn't have much funding to go back to some of the needs that these kind of trials meet. And they wanted to learn what works for whom. They also want to do this pretty rapidly. There's a lot at stake, and they're trying to get these chairs out to military vets, for example, who have these kind of disabilities. So these are the kind of standard results, efficacy terms that we see at the baseline phase. All participants just use the wheelchair using their standard way of introducing it and providing it to them. Then they're randomized to an instruction-only group or instruction plus this virtual coach reminder condition. Okay? And these are the results. I'd like for you to focus just on this mean column, if you can see that right along here. Right here. I'll just do it this way. So here we've got general discomfort. I'm not going to go into the scales, it's going to take too much time, but general discomfort is sort of like mood would be like depression, where it's a general, how uncomfortable are you feeling this week? And then there's discomfort intensity here, which is ratings of specific body parts and how painful it is in that location. And we've got frequency of using the power seat and the duration of using that power seat in large angles, meaning, how flat do they get? In the instruction group, we see that general discomfort got worse. We see that discomfort intensity got a little worse as well. And the frequency of use went down, the duration of use also went down. Okay, so instruction's clearly not where the action is. However, if we look at this virtual coach, just a simple reminder, the computer beeps and they go through this protocol. We find that general discomfort didn't change, but the frequency of use and duration of use went up quite a bit, noticeably. And discomfort intensity dropped by a whole standard deviation. It's almost half what it was during the baseline period. So this is great efficacy, right? And they've got ideas about how this works, but they'd like to be sure. And so we took the same data and we run things that are similar to structural equations modeling, but we're doing this within people. So these models are based on measures from one day to the subsequent day within people. And then we aggregate those results. I'm not going to get into the math, but if you are familiar with path diagrams or structural equations modeling, you can think of it in that manner. The point here is that they had 4 different potential models to explain why these results are happening, and their own model is the one that turned out to have the best fit to the data, thankfully. And we can take that a step further and look at, well, how does this model change depending on the intervention phase that they're in? And what we see here is that what they expected people to do, basing their PSF usage on the level of discomfort, is exactly what was happening. Over time, over the phases, so we've got baseline, instruction only, and virtual coach, you can see that relatively little changes happening here, whereas there are larger changes in those path coefficients with the virtual coach intervention. Okay, so this has wide applicability. These are just some of the areas that I've been, and studies that I've been involved with. I mentioned that I wanted to describe, just briefly mention, how these studies can be used in studying relationships. I'm working with Dr. Bayer-Merritt and Dr. Matson on studying teen dating violence over time. These are just a simple text message study. Each day, the females in the study report back what's happened in terms of a teen dating violence event and the relationship characteristics such as trust or jealousy or gift giving between them. And they're able to study what happens before, during, and after a teen dating violence event by following these paths over time. And that can be used when we're talking about relationships between mothers and babies, therapists and mothers, and so on. So this is just a quick review, an overview of the presentation. These are some references, and there's my contact information.